Good evening, everyone. So after a long time, I have come online. No. Because of your request, I am going to discuss about the amino acids and their physicochemical properties. So, whatever you want, whatever questions you are having, you can ask me anything you can type it in the chat box, and I will give answer for all the questions. Fine. So what I am going to do first is first I am going to talk about basics things. We will finish it first as fast as possible, and meanwhile you can type your questions and I will give you answer for all your questions. Fine. Will it be fine? Hi Nivans, Akshay, Anjali. So will it be fine? Am I audible? Nivans, am I audible? Good evening, Tanuja. Yeah, fine. Okay. So first we are going to say if you are going to say the components, biochemical biomolecules, we are having different types of biomolecules like carbohydrate, protein, lipids, minerals, everything are biomolecules only. Means the anything which normally forms the living cells, which supports living cells for their existence, for their survival, are called as biomolecules. If you will see all the biomolecules, proteins are the most abundant biomolecules in the living cells. If you are talking only about the living cells, then protein is the most abundant. And if I am not talking about living cells, I am talking about either living or non-living, then it is carbohydrate which is the most abundant biomolecules. So whenever you are going to get a questions like that one, which biomolecule is most abundant, then say it is asking you in the living system or it is non-living system. In the living cells, proteins. In the non-living cells, it is carbohydrate which is most abundant. These proteins, or because we are going to talk about amino acids, so amino acids ultimately gives you proteins only. This amino acids or proteins present in all parts of the cells, means because we are eukaryotes. So in eukaryotic cells, we are having different cell organelles, like we are having mitochondria, nucleus, Golgi body, ribosome, lysosomes. Number of cell organelles we are having. So whenever we are going to talk about these bio or cell organelles, so every places we are having these proteins. So either in their structural, as a structural component, or either it will be in the form of their structural component, or it will be in the form of their enzymes or the regulators or the signaling molecules. Means everywhere this protein is present in one form or in the different form, but it is present there. This proteins, without protein you cannot think about any living cells. So if protein will not be there, cells will not survive, because if protein will not there, a structure will not be there, like cell membrane will not form, different other structural component will not be there, transportation component like cell cell will not have any kind of channels, because channels are composed of proteins only. We will not have any enzymes, we will not have any regulators. So without this protein you cannot think about any cells will exist. So this protein is a fundamental basis of a structure and the function of a cell. This word came from the Greek word that is called as proteus. It means it is the first place, because it is one of the most important biomolecules in our cells. So as it is one of the most important biomolecules in our cells, so that's why from there, because it holds the first place, so the word proteus have come. And this Virgilius, he was the Swedish chemist, he suggested the name proteins for any organic compound which is most important for our life. So any organic compound which is most important for our life, that is called as protein by Berzelius. He was the Swedish chemist. After Berzelius, another scientist came. He was the Dutch chemist. He came in 1938 around, and he used the term protein for all high molecular weight nitrogen rich component which is most abundant in the animal and plant cells. So there are two different scientists we are talking about. We are talking about Berzelius, who used the term protein for all the biomolecules which is most important for the survival for the living systems, and then came Muller, who used for all high molecular weight nitrogen rich compounds. These proteins, if you see the function of proteins, it will have multiple functions, various functions. Means I have told you, think about anything and protein is required there. Any functions, like we are talking, we are eating, we are walking, we are doing anything, either we are physically active or we are not physically active. Means physically we are performing any actions, we are not performing actions. Every activity, every type of work requires proteins. So all these proteins have different type of classifications. One kind of classification for these proteins are on the basis of their role. So we have two different characteristics, two different kind of class. One class we are going to call them as a structural or a static proteins. Second class we are going to call it fixed, which does not move, which does not change their locations or concentration, that is called as static. So they are normally involved in the structural components. They form the structures, like some proteins which is present in your nail. So the protein which is present in my nail, they are not going to change their locations. The protein which is present in my hair, they are not going to change their place. Some bones are not going to change their locations. So these proteins which does not change their location, does not change their places very frequently, they are normally going to give you kind of structures, are called as structural proteins or static protein. Static means fixed protein. Second kind of proteins are which is called as dynamic proteins, whose concentration normally changes. Their location also going to be changed. Means it will be produced at one place and it may work at different locations, like pancreatic amylase, pancreatic enzymes, or different kind of protein hormones, different serum proteins, immunoglobulins. So number of other proteins, gene regulator proteins, transcription factors, number of proteins we are having. So many proteins which produced at one place and works at different locations. It goes out of the cells and then changes their location, changes their place. That we are going to call it dynamic proteins. Means they move into different places and perform their activities. Right? So examples will be for in dynamic proteins we are having hormones, we are having enzymes, blood clotting factors like serum protein I have told you, immunoglobulins, some protein which helps in the membrane fluidity, membrane transportations, then some muscle contraction proteins like actin, myosin, number of other transcription factors or enzymes, protein involved in respirations, like we are having electron transport chain proteins, different kind of complexes, those proteins. Here you need to remember one specific protein, that is alpha keratin. This alpha keratin, normally later on we will say proteins are functional only in their higher order of structures, that is called as tertiary 3D or 4-dimensional 4th degree structures. So normally proteins are functional in 3 tertiary or quaternary structures, but this alpha keratin protein, it is a alpha helix, that is secondary structure only, secondary structures, 2 degree structures, but this alpha keratin is functional effective in their secondary structure only. So that is important. So normally proteins are not functional in secondary structures, but this alpha keratin is functional in their secondary structures. Now all these proteins, whatever we are going to see it, they are composed of amino acids. These amino acids are are composed of, because it is organic compound, so it is composed of carbon, nitrogen, oxygen, hydrogen. So 5 elements. If you will see the proteins chemical constituents, chemical compositions, 5 different elements are most important. They are normally present in your proteins. They are carbon, hydrogen, oxygen, nitrogen, sulfur. These 5 carbohydrates, these 5 elements are compulsorily part of your proteins because they are present in the amino acids. We will see later on the structure of amino acid, then we will see it how it is going to be present there. Now, other than these 5 elements, there are number of other elements also may be there. Whenever you are going to do elemental analysis of your proteins or amino acids, you can see 5 compulsory elements, that is carbon, hydrogen, oxygen, nitrogen, sulfur, in different concentration, different compositions. Maximum, because it is an organic compound, so maximum concentration, maximum composition, maximum contribution is by carbon. Second most abundant compound elements is oxygen. Third most is nitrogen, because it is amino acid, so amino group will be there. Fourth is hydrogen, because it's less molecular weight will be there. This is according to molecular weight. And last one will be sulfur, because only 2 amino acid we are having which contains sulfur, so that's why it is having very less contributions. Other than that one, if proteins is going to be give you different kind of modified proteins, modified amino acids like phosphoprotein, like cytochromes, like some elements, some enzymes which requires copper, some iron binding proteins. So there we can have different types of other elements also, but that is accessory elements. These are compulsory elements, they are compulsorily part of the proteins. Is it clear? Is it clear to everyone? Yeah. So whenever, wherever you are having any kind of confusion, anything you are not understanding, you can write it down. You can write in the chat box, I will give you answer. Fine. Okay. Sure. Fine. So now if you see the amino acid, all the proteins are composed of amino acids, means proteins are the polymer. Polymer means it is composed of, it contains many amino acids are joined together and give you proteins. So proteins are the polymeric form of amino acids. Like you are having a beaded bead, you are having a necklace, beaded necklace. So every beads you can assume as a amino acids. When these amino acids joins with one thread, one bond, we are going to get a necklace. So that necklace, open necklace, is a kind of proteins. This contains, this composed of different amino acids. Now, as its name suggests, it is having 2 different word fragment. It is having amino group and it is having acid group. Amino means it is coming from ammonia. So ammonia means NH3, and acid means in organic chemistry, because I have told you this is organic compound. So organic compound means it is composed of COOH. Now, so whenever we are having any compound which is having amino group as well as acid group together in the same compound, we will call it amino acids. Fine. So if you see this compound, this is having amino group as well as acid group together in the same compound. So any compound where we are having amino group as well as carboxylic group together, we are going to call them amino acids. Wherever, I am not going to talk about place. So means it should be present on the same carbon or different carbon. I am not at all talking about that one. Wherever this will be there, location, forget about this one now. If amino group and acid group is together in the same compound anywhere, we are going to call it amino acids. Now in this case, whenever we are talking about any amino acids, so we have different forms of different amino acids. Like you can see this compound. In this compound, just for a second, in organic compound, organic chemistry, whenever we are going to have a counting, we are going to start counting of or nomenclature, the COH group gets the most priority group, that is going to give the first place during the numbering. So whenever we are going to start numbering, we will start numbering from COH group. So if you can start numbering, so it will be 1, it will be carbon number 2, and if you are having any other carbon, like R group is in third carbon, so it will be third carbon. This is if you are going to do numbering like 1, 2, 3, 4, 5. Then if you are not going to do this type of numbering, then another way of numbering is alpha, beta, gamma, delta, like that one. How to do that one? So I have told you this COH group is the most priority given group in the organic chemistry, chemistry. So the carbon which holds this COH group, the carbon where this COH group is attached, that we are going to call it alpha carbon. Like here, this COH group is associated, linked with this carbon. So this carbon holds this acid group. So this carbon I will call it alpha carbon. Correct? Now if your alpha carbon, if we are having another carbon, like this is COH group and we are having one carbon here, one carbon here, one carbon here. So this is COH group. The carbon which holds COH group, we will call it alpha, then we are having beta, then we are having gamma, then we are having delta, then we are having so on. If amino group is going to be associated with alpha carbon, we will call it alpha amino acid. If amino group is associated with beta carbon, we will call it beta amino acid. If amino group is associated with gamma carbon, we will call it gamma amino acids. So here you can see it. This is our acid group. This acid group is associated, linked with, holds present on this carbon. So this carbon is my alpha carbon, and here amino group is associated with the alpha carbon. So first you have to decide which one is alpha carbon, and then you see amino group is present on alpha carbon or beta carbon or the gamma carbon. So here first, this is my acid group. This acid group is present on this carbon, directly linked with this carbon. So this is my alpha carbon, and this amino group is associated with alpha carbon. So this is my alpha amino acid. In this case, in case of second one, we are having acid group. So this is my alpha, this is my beta, and here NH2 is associated with beta carbon. So this is my beta amino acid. Similarly, this is my COH group. This is alpha carbon, this is beta carbon, this is gamma carbon, and NH2 is associated with gamma carbon. So this is my gamma amino acid. So we have different types of amino acid: alpha, beta, gamma, delta, like that one. Now if you will say, in our body we are having many, means more than 300 plus amino acids we are having. It may be alpha type, it may be beta type, it may be gamma type, but all these amino acids does not participate in protein synthesis. Only alpha amino acid gives you proteins. So only alpha amino acids will participate in the protein synthesis, not beta, not gamma. So these alpha amino acids are called as standard amino acids, which directly involve in protein synthesis. You might be surprised why this only alpha is going to participate in protein synthesis. Anyone is having any answer? Why only alpha participate in protein synthesis? Why not beta? Why not gamma? Anyone? Any answer? No. Okay. We are having DNA. in our nucleic acid we are having message for the protein synthesis. So this DNA gives you no Anjali. So this DNA gives you messenger RNA. So message which is coded in the DNA for the protein synthesis, ultimate function of DNA is protein synthesis. So DNA gives you a messenger RNA which is having a message for which type of protein you have to synthesize. Now this messenger RNA have the message, that message will be read by transfer RNA. Now those message is response, it message is coded, it message will be read by tRNA, transfer RNA. Later on you will know that what is tRNA. So this messenger RNA is having a message which is read by tRNA, and we are having only tRNA for alpha. We don't have any transfer RNA which carry your amino acid for protein synthesis for beta and for gamma. Means we don't have any transfer RNA for beta and gamma amino acids. We are only having tRNA which will carry, no, it is mainly because of transfer RNA. Anusuya, it is Amirilal, Nivans, Anjali, Anusuya, it is because of tRNA. We are having only tRNA which can transfer alpha amino acids, not beta, not gamma. So that's why only alpha amino acids come for protein synthesis, not beta, not gamma. It may have some other usage, like you can say gamma amino acid, it is a GABA, it is a neurotransmitter. So it works in the neurons. It will have some other uses, but it will not involve in protein synthesis. Only alpha participate in protein synthesis, that is because of tRNA only. Now this amino acids, whatever alpha amino, I have told you only alpha will participate in protein synthesis. So whenever I am talking about amino acid, it means I am talking about alpha amino acid. I am not talking about beta, I am not talking about gamma. Now, so proteins are the polymers of amino acids, means it is a polymer of alpha amino acids which is joined together by a peptide bond. How this peptide bond forms? So this is your amino acid. Amino acids basic structure already you have seen it. It is having one acid group, one amino group. So and this is having some side chains like hydrogen, one R group. We will talk later on also. So and this is my this is amino acid number 1. This is my amino acid number 2. So the peptide bond is going to form because of when carboxylic acid COH group of one amino acid forms a bond with amino group of another amino acids. Means the association will be between COH group and amino group. COH group of one amino acid, amino group of another amino acids. This reactions releases a water molecule. So whenever water is going to removed, we can call it dehydration reaction. So here also it is dehydration reactions, or you can also call condensation reactions. So it is dehydration condensation reactions, and ultimately this H2O will come out and you will have a bond formation between COH group and NH2 group. So this is bond, is the bond between the CO and NH is called as peptide bond. Now remember this one. In this peptide bond, whenever you are going to write a peptide, we don't have any rule that amino group will be on your left hand side only, but by convention we are going to write like this one. Every time as a rule, as a normally conventions, we are going to write amino group on my left hand side and COH group will be on the right hand side. So this is a convention. Now this is a one amino acid. So in our body, I have told you only alpha amino acid participate in protein synthesis. So this is alpha amino acids. How it is alpha? This is COH group. This COH group is associated with this carbon, and this is my alpha carbon because this carbon holds COH group, and NH2 is associated, NH3 is associated with this alpha carbon. So this is my alpha carbon. Here also this is my alpha carbon. Now both, if you see both the alpha carbon, they are structurally looks like same, but they are not the same, they are different. How they are different? In this case, NH3 and hydrogen, NH3 is on my right hand side, in this case NH3 is on my left hand side. Now so in this case NH3 is on my right hand side, in this case NH3 is on my left hand side. So how this left right is going to affect it? It does not affect you if in normal fashion, normal manner, but if you will see in its three-dimensional structure, then you will really feel that it is a different compound. How it is different compound? Both are left and right. When it is going to rotate plane polarized light in different directions. So this rotates in different direction, this rotates in different directions. So we are having different kind of stereoisomers. So these two are different stereoisomeric compound. Now before going to see in the details, we will say any carbon, one carbon can form a tetravalency, means carbon can form 4 different bonds. So like this is 1 bond, 2 bond, 3 bond, 4 bond. So any you can call it mirror image, you can call it mirror image. So normally whenever we are talking about enantiomers, that time we will talk about mirror image. Now I am not involved in that one. I am going to tell you different things. Nivans, Akshay. So this carbon, any carbon can form 4 bonds. So this is bond number 1, this is bond number 2, this is bond number 3, this is bond number 4. This bond, for time being, here it is going to associated with a compound X, here it is going to associated with component Y, here it is associated with component Z, and here it is associated with Z, W. So one carbon can form 4 bonds. Yes, chiral atom. So I am talking about chiral atom only, Harish. So this carbon have 4 different bonds, and this carbon is associated with 4 different groups. So we are going to find that this carbon is asymmetric or chiral or not chiral. For that one, first we have to say 2 rules are there. Rule number 1, a carbon should have only single bond, no double bond, no triple bond. If carbon will have any double bond, any triple bond, means that carbon is symmetric carbon, that is chiral carbon. Means in this case you can say this carbon is having only single bond, single bond, single bond, single bond, single bond, means all single bonds, no multiple bonds, no double bond, no triple bond, means this first rule is okay with this carbon to be asymmetric. So according to this first rule, I can call this asymmetric. Now come to second rule. Second rule is whatever 4 single bond you have find out, all the groups associated with those 4 single bonds should be different. Like this X, Y, Z and W should be different. It should not be the same. If any 2 groups are same groups, similar type of groups, it means they are again symmetrical carbon. Like if I will put it hydrogen here, and here also I am going to put hydrogen, means single bond are there but 2 groups are same, here also hydrogen and here also hydrogen, it means this is same to this one. So 4 different groups are not there, it means that is not asymmetric carbon. Now so these are the 2 rules. On the basis of these 2 rules we are going to find out it is asymmetric or it is not asymmetric. So now here we are having carbon, here we are having carbon. So look at in any compound, number 1, compound number 2, it is symmetric or it is asymmetric. First, this carbon is having 4 single bond, this is single bond, single bond, single bond, single bond, means first rule is perfectly fine. Second rule, 4 groups associated with that one is same or different. This side COH group, this side amino group, this side H, this is R. So all the 4 things are different, means this carbon is asymmetric or chiral. Similarly here also 4 single bonds and all the 4 groups are different groups. So here also it is asymmetric carbon. So whenever we are having asymmetric carbon, it can give you different type of stereoisomers. So for stereoisomers to form, asymmetric carbon is necessary. If asymmetric carbon is not there, means compound will not give you any kind of stereoisomers. Fine. Now, so this is having one asymmetric carbon. So how many stereoisomers it can form? For that one we are having a formula, that is 2 to the power n. Anyone can tell me what is chiral atom? Chiral or asymmetric, what is the difference? It is same or it is different? Anyone? Can anyone will tell me? Nivans? Chiral, asymmetric carbon is only known as chiral. So asymmetric compound have another name, that is chiral carbon or chiral center you can call it. It is same thing. Either you can call it asymmetric or you can call it chiral. Clear? Question was, what is the difference between asymmetric and the chiral? It is same thing or it is different thing? So for asymmetric carbon only we are going to use a word, that is chiral center or chiral. It is same thing. So how many stereoisomers it will form? For that one you are having a formula, that 2 to the power n. So in this case we are having only one asymmetric carbon. So 2 to the power 1, it means it will have only 2 asymmetric. Yes. So it will have 2 stereoisomers. So here it is 1, here it is second stereoisomers. Now how to do nomenclature? It is which one is D, which one is L. For that one, in carbohydrates what we have seen it, that was slightly different, where there we used to say secondary alcohol, last asymmetric carbon or the last asymmetric carbon, and then we were deciding. But here it is slightly different. Here we don't have hydroxyl group. So what we will do, this is the carbon, this is the asymmetric carbon. So we will write the atomic number of all the elements which is directly, yes Harish, perfectly fine. So here this is the asymmetric carbon. So with this asymmetric carbon, whatever atom is directly associated, so we will write their atomic number, like atomic number of hydrogen is 1, atomic number of carbon is 6, atomic number of nitrogen is 7. Here also 1, 6, 7, because R can be vary, so we are not knowing here R, so we will not write any groups for R. Now if you will see here, we have to go from lower atomic number to higher atomic number. So we are going from 1 to 6 and then 7. So 1 to 6 and then 7, 1 to 6 and 6 to 7. So whenever we have, first we have to find out asymmetric carbon, then we will write the atomic number for all the elements associated directly with that asymmetric carbon. Now go from lower atomic number to the higher atomic number side. So here 1 to 6 and 6 to 7. So this type of direction. If the direction of arrow, you have started from this place and moving towards here, so if this direction is anti-clockwise, then we are going to call it levorotatory. This is anti-clockwise, means from right hand side to left hand side. So it is anti-clockwise. So whenever anti-clockwise movement will be there, we will call it L-amino acid. Whenever we are having clockwise movement or left side to right side movement, we will call it D-amino acid. So on the basis of this form, this type of pattern we are going to use in the amino acid to find out it is D form or L form of amino acids. Is it clear? So first find asymmetric center, then atoms which is directly associated with that carbon, and then you can write the atomic number and go from lower atomic number to the higher atomic number and see it is right handed side or it is moving towards left handed direction. Anyone is having any question? If you have any question, thank you. Now, so I have told you, yes, Akshay, tell me. Akshay, tell me your question. Okay, fine. So I have told you we are having 20 amino acids, 20 standard amino acids which normally participate in protein synthesis. So this is here we are having 20 amino acids. Don't go for this classifications, just see the structures. So in every amino acids, if I will zoom it little bit, I don't think. So every amino acid is having this structure is common. So one central carbon, it is same thing. Harish, it is same thing. Asymmetric carbon is called as stereo center or the chiral center. If you are having chiral asymmetric carbon, so that carbon we are going to call it, because around that carbon only we are having two stereoisomers. So the stereo center, asymmetric carbon and the chiral center are almost same thing. Okay. Now, so in all the 20 amino acids, because I have told you only alpha amino acids participate in protein synthesis. So here is the structure of 20 alpha amino acids. Every alpha amino acids, all the 20 amino acids will have a common structure. Means they are having a central carbon and they will have one side, it will have COH group, acid group, because it is amino acid, so acid group will be there. One side it is having amino group and third side it is having hydrogen. Fourth side where we are seeing R group in the previous picture, this R group, this R can be anything, it can vary from amino acid to amino acids. Like you can say in the case of glycine, R equal to H. In case of alanine, R equal to CH3. In case of valine, R equal to this propyl group. So R group is going to vary. The upper part portion is common in all the amino acids. Now, this is the common structure of 20 amino acids. So we are going to classify amino acids. So amino acids are going to be classified into different category. Like here we are having number of students. So students, I can classify all the students in different criteria. So different characteristic features. Like I can classify those who are from Punjab, those who are from out of Punjab. I can classify in national, international, Indian, non-Indian. I can classify boys, girls. So different kind of criteria might be there. So you have similarly amino acids can be classified into different pattern. So these are the 20 amino acids. So I am going to classify these 20 amino acids into different category on different patterns, different criteria. So we are having 4 important criteria. What are those criteria? First criteria is on the basis of the R group. Which type of R group is present in your amino acids? So first scheme, first criterion of classification of amino acids is on the basis of their R group. Second criteria is on the basis of their polarity of R group. R group is polar, R group is no amino acid, means on the basis of R group only amino acid is polar, amino acid is non-polar, acidic, basic, positively charged, negatively charged. Means on the basis of polarity. So second classification is on the basis of polarity of amino acid. First classification was a structure of R group. Third classification scheme is on the basis of its nutritional requirement, means it is essential, you can synthesize, your body can synthesize, or you require in the diet. So it is essential or it is non-essential. So that is nutritional essentiality. That is the third criterion. And the fourth criterion is on the basis of the fate of your amino acid. Means amino acid is going to give you, after metabolism, it will give you lipid or it will give you carbohydrate. So that is called as its fate. So on the basis of its fate, we are having 4 criteria on which we are going to classify amino acids. So first classification is on the basis of a structure. I am not at all going to show you the structure in the details. Just we will talk about what are the classes and how we are going to classify it. So first class of amino acid is where amino acids will have aliphatic side chain, means normal aliphatic side chain. So we are having 5 amino acids there. If you go to this amino acids, like here you can say we are having, this is normal aliphatic amino acids. These are glycine, alanine, valine, leucine, isoleucine. These are the 5 amino acids which is having normal aliphatic side chain. So they are 5 amino acids. They are glycine, alanine, both direction and charges are same for polar. One dot, both direction and charges. Which charge you are talking about, Harish? Harish, which direction you are talking about? For polar amino acids you are talking about, right? Harish, direction and charge. Direction, what direction you are talking about? Yes, it will be charges, means it will be varying. I will show you the different structure and then I will talk about the charges on the polar. I will talk about that one. I am coming there. Nivans, metabolic fate means if you are going to metabolize amino acids. So after metabolic process, it is going to give you carbohydrate or it is going to give you lipids. Means you have amino acids. What it is, where it is going to be utilized? In the form of amino acid, or in the form of proteins, formation of carbohydrates or formation of lipids. That is called as metabolic fate. We will come there in amino acids. Polar I am going to show you, then we will talk about, when we will tell you the polar, how polar is there, that I will tell you. So first classification is on the basis of aliphatic side chain. So 5 amino acids we are having where normal aliphatic side chain, it is normal hydrogen or methane or propane or isopropane or butane are in their structure. So that is called as aliphatic side chain. There are 3 amino acids which we are having which contains OH group, hydroxyl containing group or alcoholic group. They are serine, threonine and tyrosine. Because tyrosine I have not written here. Why? Because tyrosine is aromatic amino acid. So tyrosine we have written here. Otherwise you can write tyrosine in both places, here also, here also. This is aromatic also, this contains OH group also. So 3 amino acids which contains OH group, serine, threonine and tyrosine. If you will see here, you can see OH group, OH group, serine, threonine and tyrosine. So tyrosine is benzene ring also, OH group also. Third amino acid is, third class is which contains sulfur atom. So we are going to have only two amino acid which contains sulfur, that is cysteine and methionine. If you see the amino acid cysteine and methionine, their structures. So we are having methionine here at this place and cysteine is at this place. In this case here is the sulfur and in this case here is the sulfur. The difference between two sulfur is methionine sulfur is in between, means it is not at the terminus place, it is not at the end, it is in between the chain. So this sulfur does not participate in any other bond formations. It will not involve in any disulfide bond formation or any other reactions. So methionine does not give you any kind of disulfide bond or any other reaction because sulfur is in between. This is called as thioester. Thioester means, thio means sulfur, ester means like this type of bond. So this is called as thioester. Whereas cysteine is having terminus, sulfur is present at the terminus at the end. So it is called as sulfhydryl group. It is like OH group. So it is sulfhydryl group. So methionine have sulfur in the form of thioester, which is less reactive, which does not participate in disulfide bond formation or any other chemical reactions, whereas cysteine have terminal sulfur where it involves in different kind of bond formation as well as the disulfide bond formation. This is called as sulfhydryl group. Yes Harish, same thing, all the three. This is cysteine. If we will write cysteine, it means two molecule of cysteine joins with disulfide bonds and give you cysteine. So difference with this E, Te, that E, if you will write it, it means single amino acid. If you are not going to write that E, then it means associated with disulfide bond. Fourth group is acidic amino acids. So there are two acidic amino acids we are having, that is aspartic acids and glutamic acid. So aspartic acid is going to be linked with amino group, so it is called as amino derivative of aspartic acid, that is called as asparagine. Amino derivative of glutamic acid called as glutamine. So this is acidic amino acids and this is their amino derivative. So aspartic acids amino derivative is asparagine. Amino derivative of glutamic acid is called as glutamine. Like if you see structures here, this is here we are having aspartic acid, here we are having glutamic acid, here we are having COO, here also we are having COO. If one of the oxygen is replaced with amino group, like here, it is called as amino derivative of aspartic acid, aspartate. It is called as asparagine, amino derivative. Similarly, glutamic acid is going to be one of the oxygen is replaced with amino group. It is amino derivative of glutamate or glutamic acid. It is called as glutamine. So we are having four amino acids in that category where acids or acid derivatives. Next class is basic amino acids, which is having extra NH2 group. One extra acid group was there, so that was acidic group, acidic amino acids. If amino acids have one extra NH2 group, amino group, it is called as basic amino acids. There are three amino acids which contains extra amino groups like lysine, arginine and histidine. So these are the three amino acids which contains one extra amino group, like one naturally it is having, one extra it is having. This is already every amino acid is having. These two are the extra amino groups they have. Similarly, this is naturally they have. These are the extra amino group they are having. So this is called basic amino acids. So we have three amino acids which is basic in nature, that is lysine, arginine and histidine. Then we are having amino acid which contains aromatic ring, aromatic benzene ring. There are 3 amino acids which contains aromatic benzene ring, that is phenylalanine, tyrosine and tryptophan. So these 3 amino acids contain benzene ring. Remember again, this tyrosine, this is not only aromatic but it also contains OH group. So tyrosine you can place at 2 different locations, 2 different places. And the last 7th class is amino acid, which is not the amino acid, which is called amino acid, that is proline. This one, in this case we don't have a natural normal structure like this one. In this case, in case of proline, the amino group and the R group forms a ring structure. Like you can see, this is amino group and this is R group, so they forms a ring like a structure. So it is not the natural amino structure, amino group, amino acid, it is called like amino acids. So out of 20, 19 amino acids, 1 amino acids. So what we have seen, total 7 classes on the basis of the structure of R groups. First class, aliphatic side chains, 5 amino acids there, glycine, alanine, valine, leucine, isoleucines. Amino acid which contains OH group, 3 amino acids there, serine, threonine and tyrosine. They are hydroxyl containing amino acids. Amino acid which contains sulfur, 2 amino acids, one is thioester group, that is methionine, other one is sulfhydryl containing group, that is cysteine. Fourth class, acid or acidic derivatives, that is aspartic acid, asparagine, glutamic acid, glutamine. Fifth class, basic amino acids, that is lysine, arginine and histidine. Sixth class, aromatic amino acids, phenylalanine, tyrosine, tryptophan, and last amino acid, that is proline. So this is one classification, one type of classification. Second type of classification is on the basis of the polarity, which Harish was talking about. In this case, we have 4 classes, 4 type of classes. First class is of non-polar, second class is of polar, third class is positively charged, and fourth class is negatively charged. We know whenever we are going to write amino group, so amino group is basic groups and basic group can take extra protons, so it will have a positive charge. Means whenever we are talking about any amino acid which is having a positively charged, it means we are talking about the basic amino acid. So in previous classification we have seen we had 3 basic amino acids, that was arginine, histidine and lysine. Lysine, arginine, histidine. So these 3 basic amino acids are positively charged amino acids. 2 amino acids which had acidic groups, they have extra acid groups, means they are negatively charged, like aspartic acid and glutamic acid, only 2 amino acids we are having, so those 2 are negatively charged. Then we are having polar groups. Polar means which is water soluble. So 3 amino acids which is which contains hydroxyl group, that is serine, threonine and tyrosine, plus 2 amino derivative of aspartic acid and glutamic acid. So 5 amino acids, 3 OH containing amino acids, 1 amino derivative of aspartic acid, 1 amino derivative of glutamic acid. So 5 amino acids. So 3, 2 and 5. So 10 amino acids we have divided in these 3 classes. Remaining 10 amino acids will be non-polar. So no need to remember these non-polar amino acids. Remember this basic amino acid, remember this acidic amino acids, remember this polar amino acids. Automatically remaining will come in this non-polar group. Is it clear? Any question? Nivans, Harish, Akshay, Anushya, Sheetal, Dorji, anyone is having any question? Achal, Anjali, anyone is having any question? So in this classification you need to remember remaining 3. You remember this one because only 3 amino acid will be there, only 3 basic amino acid will be there. Base normally positively charged, acid normally negatively charged. So remember this one, 3 OH containing amino acid here, and the amino derivative of aspartic acid and glutamic acid also here. So remember this one, and this one, automatically remaining will come here. Now this was my second classification scheme. Third classification scheme is on the basis of nutritional essentiality. Means it is required by your body. Every amino acid, 20 amino acid we require for protein synthesis, but does it synthesized by your body or not synthesized by your body? Because we require it. So either I have to synthesize directly in my body or I have to get it from the external sources. So anything which your body requires and you are able to synthesize directly whenever your body needs it, they are called as non-essential. Why we are going to call it non-essential? Because whenever body requires, body can synthesize automatically by themselves itself. So it is non-essential. We don't require we are not depending on anyone else. For those compounds, those amino acids, those components which your body requires but you are not able to synthesize by yourself, for that one you depends on others, you depend on external sources. They are essential components. That should be essentially part of your food. Essentially it should, you have to take in your diet. Similarly in the amino acids also, all the 20 amino acids we are having, 10 amino acids your body can synthesize whenever they require. So 10 amino acids are called as non-essential amino acids. Those 10 amino acids which your body can synthesize themselves according to their wish whenever they require, then whenever they need. So they are non-essential. But there are 10 amino acids which your body cannot synthesize by themselves. Whenever body needs, body requires it, you have to provide them. Either previously you have given them, so it will be in the stored form, or you have to supplement it either in the form of tablet or in the form of diet, in the form of different dosage. So that is called as essential. So here is the list of 10 essential amino acids. So this is 10 essential amino acids which your body cannot synthesize according to their wishes, according to their requirement. So this should be part of your diet. How to remember this one? So if you are going to remember essential, remaining 10 will be non-essential. So remember either 10 essential or remember 10 non-essential. So we have two formula. Either remember this one or remember this one. This is sequentially written there, like M for methionine, A for arginine, T for threonine, T for tryptophan, V for valine, I for isoleucines, L for leucine, P for phenylalanine, H for histidine, Ly for lysine. This is Matt will fly or private team hall. Whatever you want, you can remember it. Private team hall, phenylalanine, valine, threonine, tyrosine, isoleucine, methionine, histidine, arginine, leucine, lysine. Now, so this 10 amino acids are essential. Remaining 10 will be non-essential. Clear? Now, so this 10 amino acids, these are non-essential amino acids because your body can synthesize whenever they require, whenever they needs it. Now, in this 10 essential amino acids, whatever we have seen it, 2 amino acids, that is arginine and histidine, they are called as semi-essential amino acids. So arginine and histidine, which we have classified as essential amino acids, they are called as semi-essential amino acids. Why it is semi-essential amino acids? Because in the childhood, whenever baby develops, baby grows, that time it is essential. That time baby cannot synthesize it. So we have to provide them in their diet. So in the childhood it is essential, but when we are going to be adult, that time we can synthesize by ourselves according to our requirement. So in the adult it will be non-essential. Histidine and arginine are essential for child's, for baby, for growing age. Whenever person will be adult, it becomes non-essential. So because its nature is going to change, become essential and non-essential, so we are going to call them semi-essential. In one phase of our life it is essential, in another phase of our life it is non-essential. So it is called a semi-essential amino acids. Fourth classification scheme is on the basis of its fate, means after metabolic process it is going to give you carbohydrate or the lipids. So we are going to call them ketogenic if it is going to give you lipid or ketone. So either it will be ketogenic, or if it is going to give you glucose or carbohydrate, it is called glucogenic. So we have three categories. Either it will be pure ketogenic, means it will give you only lipids. In the growing age, till we are going to grow, that time you can call it around 20-25, because that till that time we are going to grow, we need development. So till that time it is normally essential, and after growing, once we are going to be a adult one, maybe might be there are some roles of some of the adult hormones, it might be, we are not sure, but might be there are some roles of the adult hormones, like sex hormones you can call it, they might have some roles. So on the basis of fate we have three categories. Either it will be ketogenic, or it will be glucogenic, or it will be both ketogenic and glucogenic. Now whenever we are talking about ketogenic, means it is never will be glucogenic, it will never give you carbohydrate, it will only give you lipids. So only one amino acid is there, that is leucine. So leucine is the only amino acid which is pure ketogenic, it will only give you ketone body or the lipids. There are 5 amino acids which is both ketogenic and glucogenic, means it will give you both lipids and ketone. They are lysine, isoleucine, phenylalanine, tyrosine and tryptophan. So 1 is here and 5 here, so total 6 we have used in this 2 category. Remaining is 14. Those remaining 14s are pure glucogenic, means those 14 amino acids will not give you lipids in any conditions. Means leucine only lipids, no carbohydrates. Lysine, isoleucines, phenylalanine, tyrosine and tryptophan, both carbohydrate and the lipids, and remaining 14s are only carbohydrate, never it will give you lipids. So this is 4 different classification schemes of amino acids. Any questions in the basics of amino acids? Any questions in the classification of amino acids? If you are having, please ask. Any questions? Shall we continue? Like whenever we are having body requires protein, like I have got infections, I have got a kind of parasitic disease, so that time my body requires some kind of serum proteins, immunoglobulin synthesis, means your body requires protein synthesis, so automatically amino acid synthesis will start. So we are having synthetic machinery synthetic process. So we can synthesize, we are having only metabolic process, synthetic process for non-essential amino acid, and we are going to call it non-essential because we synthesize it. जो चीज़ हम घर में बना लेते हैं ना उसको हम नॉन इसेंशियल बोलते हैं, वो हमारे लिए जरूरी नहीं है. जो हम नहीं बना पाते हैं, जो हमें बाहर से खरीदना पड़ता है, दैट इज इसेंशियल फॉर मी. Because isolation will give you both ketone lipid synthesis, lipids as well as ketone lipids or ketone body you can call it, and it will also involve in carbohydrate synthesis, glucose synthesis through gluconeogenesis process. So it involves in both the processes, both the places. So that's why if you see the leucines structure and isoleucines structure, they are slightly different. Their carbon chain is R group, carbon chain is different. So because of that carbon chain difference, this is involved in leucine sorry. So because of R group differences, leucine is only ketogenic, whereas isoleucine is both ketogenic and glucogenic because of that difference in their R group, means carbon chain only. Shall we continue or stop? Any other question anyone is having? What? Shall we stop or continue? Harish, have you got your answer? Say, Nivans, whenever we are having body requires, like body in the growing phase, like we grow, in our life we are going to divide into 3 phases. This is baby phase, adult and old age. In the baby, more protein synthesis will be there. So we are going to have more proteins synthesis than the degradation. For this protein synthesis in the baby phase, in the growing phase, we require more protein synthesis. More protein synthesis means we require more amino acids. So here amino acid synthesis will be more. Not only in the case of this one, we are going to have more protein synthesis, so more non-essential amino acids by our body and essential through the diet. Not only for the growing phase, but for disease also, for other processes also, other things also. Fine. Similarly, at the adult age, the synthesis of amino acids and the degradation of amino acids is almost same. So we don't have either aging or we don't have any growing, means neither we grow nor we deteriorate. So here synthesis and or in this case degradation will be more, synthesis will be less. So this is just one thing I am talking about. For growth we require protein synthesis. Not only growth, second thing I am telling you, whenever we are having disease, in the disease case we require more protein synthesis to fight with the disease. For we are going to have any kind of infection, we require having any kind of wounds. To fill that wounds, we require more protein synthesis. So in this case, those cases, body synthesizes more proteins. For every cases wherever we require more protein synthesis, we require more amino acids. So we have 2 types of amino acids. Either it is essential or it is non-essential. That is on the basis of your body can synthesize or cannot synthesize. Those amino acids which your body can synthesize, we are classifying them into non-essential category, because मेरी जरूरत की चीजें हैं मैं बना लूंगा वेन एवर बॉडी रिक्वायर्स इट. Essentials are those which your body cannot synthesize it. You require, and in these cases, essential also, but your body cannot synthesize it. So that time you have to provide them from the external sources. That's why protein deficiency disease, amino acid deficiency disease mostly occurs in the kids, not in the old age, because in the growing phase we require more proteins, more amino acids. Is it clear? Okay, fine, no issue. Tomorrow 6 o'clock. Tomorrow 6 o'clock we will talk about physico-chemical properties of amino acids. Okay, so whenever you are having read it, try to watch it, try to read your textbook. Whenever you are having any issue, any confusion, any problem, just write in the notebooks, and then when you are coming in the class, that time you can ask me. Fine. Or you can send me on WhatsApp also. You can ask me on my WhatsApp also. Not only this part, amino acids, in anywhere in your biochemistry, whenever you are having any issue, you can ask me anytime. I will give you answer. Fine. So thank you. So thank you very much. Thank you everyone.